This video is on the relatedness of all living things and is part of the evolution of organisms topic. So the key concepts from the subject outline look at mutations and how they arise and how they accumulate, looks at other sources of variation, especially those involved in sexual reproduction, and then how we can analyze DNA differences to look at relatedness. So one way to think about relatedness is to look at this family tree of human languages. So we can see here the leaves are current existing languages, and the brown stems there representing where they came from. So French and Spanish are closely related, and English and German are closely related to each other, but also we can connect French, Spanish, English and German back through a route here that goes back to the Indo-European languages. But when we look at these languages, we can see that they have a common alphabet, share some similar grammar and some similar words. But over here we have the Sino-Tibetan languages, including Mandarin Chinese and Burmese and Thai. These are completely separate to the Indo-European languages. They don't have any of the similar alphabet or words or grammar structures. And so this is fairly good evidence that these languages arose in different areas at different times independently. So let's consider how life arose and looking at different organisms today. We certainly know that animals and plants and fungi and bacteria are quite different to each other, and it may be possible to think about conjecture that they arose separately at different times in different areas and that life arose multiple times on the earth. However, the evidence doesn't suggest that. The evidence suggests a model much more like this, that all organisms have arisen from a single ancestor. The key evidence behind this is DNA, the language of life. That language is absolutely the same in all living things that we've found so far. Exactly the same chemical alphabet of A's, T's, C's and G's. Also the genetic code of how those then translate into the amino acids to make proteins is the same in all living things. So this is excellent evidence that all living things arose from a common ancestor. Just for the record, this ancestor is known as Luca, the last universal common ancestor, from which bacteria, archaea, which are another form of single-celled life, as well as the eukaryotes, that includes animals, fungi and plants. So even though we had a single ancestor, clearly we're not all the same as each other. So how do these differences arrive? Where does variation come from? So the key thing is to go back to our understanding from DNA and proteins that there are mutations that change DNA, either through copying errors like this or through damage to the DNA, and that these changes accumulate over time. So if we look at an example of a short DNA sequence here in generation 1, a mutation from G to C might occur somewhere between the first and the 10th generation. That is then passed on to subsequent generations and another mutation may arise. So as we keep going down through these generations, we keep on getting more and more mutations accumulating, so that when it's distantly related, 40 generations ago, there are 4 changes between our DNA at the end and the DNA at the start, whereas more closely related generations have fewer differences between them. One way we can measure differences experimentally between the DNA of different organisms is by a process called hybridization. So here we have DNA from organism A and DNA from organism B. If we heat that to separate it, just like in PCR, and then cool it so that those strands come together, if they were matching sequence exactly all along, then there'd be pairs of complementary single strands which would bind to each other perfectly like this. If there were sequence mismatches or differences, then they might partially hybridize and other parts wouldn't join. And if these two sequences shared no similarity, there'd be no hybridization. So it's a very quick and easy way to compare DNA of 2 different species, 2 different samples, to see if they perfectly match, meaning they're closely related, partly match, partly related, or don't match at all, which means a very distant relationship. Another way to do this with modern sequencing technology is just to sequence the DNA like we saw before. So here we have the DNA from a gamma globin protein in the DNA here, which is commonly found in these different species. good luck if you can find out which ones they are, just by guessing. Obviously Homo sapiens should be fairly obvious for most people, and gorilla, that's another obvious one. Pan troglodytes is a chimpanzee, and Pongo pygmaeus is an orangutan. And we can see the closely related DNA sequences between these, which is strong evidence that these species are also closely related. A final way to compare relatedness is then to take it one step further and to look at amino acid sequences. So whereas DNA sequences like this can change fairly rapidly, but some of these DNA sequences might not actually make a difference to the amino acid that's there. Especially, this is as part of a protein, but lots of DNA sequences aren't, and so they can change without having much of an effect on the organism. Whereas here we're looking at amino acid sequences, and in particular this is in hemoglobin. So you can imagine if hemoglobin changes, that's the one that carries oxygen around the blood, it's not going to be very good. You don't want a lot of mutations in that. So the hemoglobin protein amino acid sequence changes very slowly over time. And this allows us to compare the differences between human and macaques, only 8 amino acids, humans and dogs 32, birds 45, frogs 67, and lampreys 125 amino acids are different in that protein. So even though all of these species have red blood cells with hemoglobin protein, those proteins have up to 125 amino acids different. And clearly, if you have fewer differences, it's most likely that you had the same DNA most recently a few generations ago. Whereas back here we're talking thousands and thousands of generations ago. So one way in which we can map out these differences and try and look at the relatedness of species was shown in that previous diagram a little bit. It's called a phylogenetic tree. So here we have time in this direction, the distant past down here and the present time up here, and some sort of variation, whether it's an amino acid sequence or the length of a tail or the color of a wing. There's some variation that you're measuring in this distance. You look at the existing organism. Here's where it sits on the variation. And of course, when you take one organism, an individual, that's part of a population, and you could have a bit of variation within that population, but that one stands as a representative of that population. You look at another couple of organisms that might be in a different population, and you can see where they sit on the variation scale. We might think of these perhaps as 3 different species, and with representative individuals from each. If you look back at time and find a couple of individuals with some other sort of level of variation in terms of perhaps the length of the femur, we might put them here. We know that this bone was around this time and has this length, this bone was at that time and has that length, and an even older one has this. So the job of us here as archaeologists or classifying or trying to work out the evolutionary scale here is to find out how is it that these populations with these different lengths arose, and how might we put these together to form a family tree. Regarding, of course, that each of these is part of a population as well. So there's some sort of variation here, but we think that these ones go together and these ones go together. It makes sense that the ones of a similar length probably arose from ancestors of that similar length. So there are 2 different models for how we can do this. One is called gradualism. This one assumes that the changes, perhaps again back to that femur length, don't really change that rapidly, and we can gradually move from one to the other form. The other theory is around punctuated equilibrium. This one says that most populations, that femur length, for example, will stay the same for many generations, and then suddenly something happens that means that shorter or taller animals are favoured, and then we suddenly have a divergence of the population into the ones with the short legs and the ones with the long legs. So when we look at those 2 models, the gradualism looks like this. This femur's slowly getting shorter, this one's slowly getting longer. In the punctuated equilibrium, we see suddenly there's a point where short or long is good, but not in between, and that's where the population diverges. So it's interesting to think about this in terms of how we trace back these trees and thinking about human evolution. When we look at these sort of pictures, we often have the false view that humans evolved from chimps. And this is a very common, popular thing that's said that humans evolved from monkeys or apes, and it's absolutely wrong and not true. When we look at chimps and other apes, they are the same as those points on the top of the tree there. So what we're talking about here is that chimpanzees and Homo sapiens both arose from a common ancestor. So it's not that this turns into this, it's that we would have had some common ancestor down here which evolved towards being a chimpanzee, and other ones here that arose to being all these different types of hominins or human ancestors, hominids, that we are aware of, including Neanderthals. Now, Neanderthals are a really interesting case study. If you want to look at some of the extra information on those, I recommend it. Homo sapiens and Homo neanderthalis were around at the same time, about 40,000 years ago. Coexisted, would have met each other. So it's not that one evolved into the other, they both co-evolved from this common ancestor here. And so it's a fascinating thought to think about two human-like species coexisting at the same time, and how, if Neanderthals were still around, how we'd interact with them and work with them these days.